We'll get started on cells today. These illustrations are from OpenStax, which is an online source, open source for textbooks and help in all different topics. So today we're talking about cells. We're going to have a lot of different illustrations. These will be different than the illustrations in your textbook, but that's actually a benefit. You want to see these ideas and concepts in different formats. You don't necessarily want to memorize a picture. You want to understand what you're looking at, be able to recognize a Golgi body, whether it's in one picture or in a different picture or on a model, in a classroom, in a diagram, in a qualifying exam you might be taking in the future. So all living things are made of cells. All living things are made of cells. Cells have a plasma membrane, and cells arise from other cells. In this illustration, we're looking at a cross-section of some plant tissue. We're seeing some plant vascular tissue, and mostly we're seeing the cell wall. Not all cells have a cell wall. A wall is more rigid than a plasma membrane, but all cells do have a plasma membrane. So remember to keep those straight. They seem kind of similar, right? So a plant cell has both a plasma membrane and the cell wall. Animal cells do not have a cell wall. We wouldn't be able to move around if we had cell walls. So here in this illustration, you're looking at the cell walls of plant material, and the rest has been removed in the preparation. This was taken with a light micrograph. Let's also remember that they're the smallest part of a living thing that is can still be alive. It's easy to get confused between atoms and molecules because we've been talking about all things are made of atoms. So that's all things. So a cell is made of atoms and molecules, but we can get down to a given atom or molecule. That's not alive. That's a building block of matter, and we look at that from the chemistry side. All living things are made of cells. That's the smallest unit, smallest functional unit of life. In this illustration, we see cheek cells from a human. So there we have a nucleus, right? There's the nucleus. And we see the entire cell. This was taken from inside someone's mouth, just gently scraping the inside of the cheek. And then a stain was used to make it show up. And if we were in face-to-face lab, you would do this yourself, and you could see these cheek cells. And you can clearly see the nucleus. We can almost make out some mitochondria there. You can see some light spots in this one that may be some other organelles, but we clearly see the nucleus. Nucleus is a characteristic of eukaryotic cells. We'll talk about that more in a minute. All right. Cells are the building blocks of all organisms. So remember, organisms is not necessarily, if it's matter, it's not necessarily an organism. In single-celled organism, the cell is the entire organism. But we're talking mostly in human biology, we're talking about human cells. So there we have some animal cells, some plant cells, and looks like we've got some bacteria there. Right? These two illustrations were taken with a light microscope, and a stain was used to make those cells come up. If we were looking at these under the light microscope without a stain, the outlines will be very, very faint indeed. And we do see here the plant cell walls. This was taken with a scanning electron microscope, and the color was added later. The photographs that we get from scanning electron and transmission electron microscopes come in black and white, but often the books will add color so that we can differentiate. So remember, when you're looking at the illustrations in the book, that those colors are added to accentuate the different organelles, to help us distinguish one from the other. If we were looking at something in black and white, as learners, we wouldn't know what it is. Later on, you'll be able to distinguish rough ER, right, from a transmission electron micrograph. But at the beginning, we don't know what that is. So that's why we have all those different colors in the book. The hierarchy, or levels of organization, should seem familiar because we covered them in Chapter 1. So cells are the basic unit of life. That's as small as we can go and still have something that's alive. If we go smaller, we'll have atoms and molecules, and those are not alive. Next up from cells, we have tissues, and tissues are composed of interconnected cells with a common function. Those cells could differ from one another. And then several tissues can combine to form an organ. Examples of an organ would be the stomach, the lungs, the heart, a particular bone, your eye. Organs working together make up an organ system. In our textbook, we use the organ systems as chapters. So we'll look at respiratory system, nervous system, digestive system. Of course, these organ systems work together, and it's important that you understand how these systems work together. But let's start to worry about that part towards the end of the class. Once we know enough about each organ system, it'll make more sense to see how they work together. So an example of an organ system would be the respiratory system, and that would include the nose, the pharynx, the windpipe, the trachea, and the lungs themselves. And of course, multiple organ systems together form the organism. So an organism is one individual of whatever species you're considering, in this case, human. Cell sizes can vary, and most are too small to be seen by the naked eye. So we've got some microscopes here. This is a light microscope, similar to what you would have in lab. And this is a transmission electron microscope that would be at larger universities. These are very expensive and require a strong foundation underneath. And if your institution offers a course in scanning or transmission electron microscopy, consider taking it. It's a great thing to have on your resume. All right, magnification and resolving power. So magnification is how big something is, but resolution is how clearly we see the details. So think of a high resolution TV, high definition TV, that's resolution, right? So in this picture, we see some diatoms with excellent resolution. This was taken with a light microscope. Here we see magnification of the same organism. So we can tell that this is a butterfly, and here we get a closer view, and here we can even see the mouthpiece and the eyes. That's magnification, is getting bigger. Okay. Resolution is seeing something more clearly. So here we see this organism under a light microscope and under a scanning electron microscope. Here we can see the resolution of this surface shape much better with the scanning electron microscope. An advantage of the light microscope is you can observe things while they're still alive. In transmission, you cannot. All right, so resolution we can think of as high definition or better clarity of the detail. We can get the detail. You notice that we've got a similar size, right, in both microscopes, but scanning, our big advantage is that surface definition, and that's what makes scanning electron micrographs so interesting to look at. You can find many online. When we're using a light microscope, we use a stain, we use chemical stains to make those cells show up. If we're looking at plant cells, we can see them because the cell walls are a little bit thicker and the chloroplast is green. And so we can make that out, and we can see the cytoplasmic streaming if we're looking at Elodea cells. But in animal cells, the organelles themselves really don't have any color. So in this illustration, we can just barely make out the plasma membrane and the nucleus. In this illustration, we can see them a little bit better, and we've used a stain to make them show up. There's the nucleus, there's our plasma membrane, and you can tell that there is some additional structure in the cytoplasm. We can't really make out what it is with the light microscope. And here we've used a stain to get a better view. We can see the nucleus in the middle, we can see the plasma membrane, and we can again see that there is some structure in the cytoplasm that's difficult to make out. So this is what we would see if we were in face-to-face lab. We can take samples of our cheek cells. We can do this without any injury and easily stain them and look at them in class. Cells can vary a great deal in size. So we were just looking at cheek cells, and they would be about here, like animal cells. All right, and many plant cells also here. Pollen a little bit larger, human egg larger, and then frog egg larger, and then of course hen egg would be out here somewhere, chicken egg. Red blood cell, a little smaller than other animal cells. There's mitochondria, bacteria, viruses are yet smaller, and then we have protein molecules, lipids, and down to atoms. So we're right in here, right? Red blood cells don't have all the organelles, so they are a little smaller. They also need to fit through the capillaries easily. Here we see an infection. So these cells are white blood cells from a human. So this was extracted from cerebrospinal fluid, and there are rod-shaped bacteria in it. So these rectangular cells are bacteria. These are human white blood cells, and we'll learn more about them when we get to blood. So this was a sample taken from someone with an infection in the cerebrospinal fluid. All right, here we see a different bacteria that causes Lyme disease, and here we can see the shape is kind of a spiral or wavy shape. So among the bacterial cells, there are different shapes, and we can also see in these unfortunate illustrations the difference in size, right, between human cells and the bacterial cells. Bacterial cells are smaller. They also do not have a nucleus. All right, here is a closer look at some of these common bacterial shapes. We will learn more about those in microbiology, which you'll probably take in the future. So we've got round, we've got the bacillus, or rod-shaped, and then we have some various spiral or curved shape. Here's our microscope. Normally we would be looking at the microscope in face-to-face lab. I will tell you, when you go to pick this up, you're going to have one hand appear on the arm or the body. You're going to have another hand underneath. You're going to support it from underneath, because the old style microscopes, the bottom can fall off. If you just hold it up here, the bottom can slide right off. And there's nothing wrong with an old microscope. If you're working in a doctor's office and maybe she purchased that microscope years ago, or she's using one from her father's practice, you want to support it from the bottom, right? Anytime you're moving a microscope, get in the habit of supporting it from the bottom, because there's nothing wrong with an old microscope. It's still effective. If you're in class, most of the educational microscopes are meant so that they can't be damaged easily, but you want to get in the habit of handling them properly. We've got the eyepiece or ocular up here, and these you can adjust side to side to fit your own eyes. and there will be a number there between them that shows you that distance. Once you know that distance, that will be the same on any microscope. For me, I know that that's 61. So if I were in face-to-face lab and you asked me to come look at what you've got under the microscope, I'm not even going to look until I've moved those eyepieces to fit the width of my eyes, and then I'll look. So those are the oculars or eyepiece, and there's a lens there. These are the objectives, and there's a magnifying lens in here as well. Often, but not always, the eyepiece objective is going to be 10 times. And sometimes these will lift out, right? Sometimes these eyepieces will lift out. Usually in a classroom microscope they will not, but in a research microscope, or something you find in a lab, you can often lift those out and put in a different magnification. But 10 is common. And then you have the objective lenses, and this is a revolving nose piece that spins around. When you're switching to another objective, you want to hear it snap in. You want to make sure it's snapped in. You're always going to start on the lowest power, and that's called scanning. And don't get confused with scanning electron. So that smallest objective is called scanning because it's going to give you the widest field of view, and it will allow you to scan that slide and see more of the tissue and try to find what you're looking for. The next objective, and that's often about 4, magnification of 4. So if you've got it on the scanning and you've got the 10 in the ocular, your total magnification is going to be 40. You're going to multiply your ocular times your objective lens. Your next step up may be 10 for low power, and often we'll find exactly what we want to look at on low, and then our next up would be 40x objective for a total of 400. Depending on what class you're in, that highest may be an oil lens. Make sure you get clear on how to use that oil lens with your instructor before you use it. You can damage the lens. You only want to use that oil on the oil high power. The highest power without the oil is called high dry or high power. So dry means without the oil. When you're cleaning your microscope, only use lens paper. Only use lens paper. Do not use a tissue. Don't use a paper towel. Don't use a Kim wipe. You're only going to use lens tissue. All right. It's hard to make out on this slide, but there's a mechanical stage here that allows you to move that microscope slide a little bit. Right? So this thing is called the stage, and there's a clip on there that holds the slide. When you're placing that microscope slide onto the stage, lay it flat. Lay it flat on the stage, and that mechanical stage should have a little spring type lever. When you fit that slide into that lever, lay it flat. Do not jam the slide underneath that lever. That lever has a spring on it, and it will gently hold that slide in place. So everything should be flat. No jamming underneath the lever. Okay? There's a little spring in there that lets that lever come back and gently hold that slide in place. And then you've got some knobs hanging down. There's your knobs hanging down. Just get used to those, play with them until you're familiar with them. And that will move the slide around so that you can see different parts of the tissue on the stage. These are your coarse focus and fine focus. I call them focus. Sometimes you'll see these called adjustment. So you've got coarse focus, which is going to be a larger knob, fine focus, which is smaller. All right, we talked about this X and Y stage knobs. You're just going to have to practice with those. You've got a rheostat or a dimmer switch that will adjust the amount of light. You've got your lamp. They're calling it illuminator here. We've got the condenser, which pulls that light in towards your slide. And then you've got your diaphragm or iris diaphragm, which can adjust that light. So as you're becoming familiar with the microscope, try moving that condenser and getting familiar with it. There also should be an on-off switch somewhere for your light. So when we go to focus, we've got our slide on here. We've maybe held it up to the light and seen the tissue, and we put that right in that circle of light right here. You've got your objective on your lowest power, and you're not looking through the microscope yet. You're going to tilt to the side and look through, and you're going to use that coarse focus to lift that up all the way. You're going to use the coarse focus to pull that stage up as far as it'll go. All right, you haven't even bothered to look through yet. Now you can look through, make sure that you've got the area of tissue that you want to see, and then you're going to slowly use the fine focus, that small knob, to move it down. And that blurry image should come into sharp focus. If you have nearsighted or farsightedness, you will automatically correct for that. So if you're nearsighted or farsighted, just take your glasses off and use the microscope. You will correct for that. And if you've got astigmatism, you're going to have to use your glasses. If you're using glasses for using the microscope for a long time and you have astigmatism, you may want to consider getting a contact lens. All right. Electron microscope. So with the electron microscopes, we can see higher magnification and higher resolution. In this illustration, we have fabulous resolution of a mitochondria. And even though I haven't seen this particular picture before, we know that it's a mitochondria because of the shape and these internal membranes. Fabulous picture. This is transmission, right? This is transmission right here. This is scanning electron microscopy of red blood cells, where we're getting a view of the surface. So this is a much higher magnification. It's giving us excellent resolution. This scanning electron micrograph is also giving us excellent resolution, and we can, well, no, I'll just tell you now, that little dimple is on both sides of the red blood cell. You may see similar illustrations with the cell in red. They've added that color afterwards. So, very good. Look for similar illustrations online. There's lots and lots of fun illustrations to look at from electron microscopes. So, in review, cell theory is one of our underlying principles of biology. All living things are made of cells. All living things are made of cells. Cells are the basic units of life, and all cells come from pre-existing cells. All cells come from pre-existing cells, right? Okay, see you in the next mini lecture.